Exocytosis is the transporting of unneeded molecules out of the cell. In exocytosis, the vesicle fuses with the plasma membrane in order to remove unneeded molecules from the cell. This requires the use of energy, or ATP. Unlike exocytosis, endocytosis is used when transporting molecules into a cell. This occurs when the plasma membrane pinches in to form a vesicle, which requires the use of ATP. Different types of endocytosis include phagocytosis endocytosis, pinocytosis endocytosis, and receptor endocytosis. Phagocytosis endocytosis is when a solid substance or a large substance is brought into the cell by endocytosis. This is essentially cell eating. Pinocytosis is when a liquid or a solution is brought into the cell by use of endocytosis. This is essentially cell drinking. Receptor endocytosis is when molecules bind to specific receptors on the cell membrane, and then the membrane pinches in by endocytosis and forms a vesicle. The sodium-potassium pump is particularly relevant in neurons. In these nerve cells, sodium ions from the inside of the cell are bound to the carrier protein. ATP then flips this protein, and sodium ions are released to the outside of the cell. When the sodium ions are released to the outside of the cell, potassium ions from the outside of the cell are then bound to the carrier protein, in which ATP again flips the protein, and potassium ions are released to the inside of the cell. This process creates a net 1 charge on the outside of the cell because there are more sodium ions on the outside and more potassium ions on the inside.